It's a pleasure to be here and to talk a little bit about regenerative medicine. Jeff's about to pull up my slides. I decided on a title, Regenerative Medicine, the Hope, the Hype, and the Future. I guess I could have used 25 years of progress, but still only a beginning, because I think it still is only a beginning. And I'll try to give you a historical perspective and also where I think we are right now. So, starting with the historical perspective, even prior to 1950, there were references to the concept of tissue engineering in the literature. In the 70s and 80s, research on what we could now call tissue engineering emerged. And the term tissue engineering was coined in 1987 at a NSF meeting in Washington, D.C. Regenerative medicine, that term began to be used in the mid-90s. There's some who think regenerative medicine is simply stem cells, but it's far more than that. And in fact, whatever term you want to use, what we're talking about is the replacement, the repair, and the regeneration of tissues and organs. I put this up here just to show that there were visionary thinkers way back when. This is a book called The Culture of Organs, written by Alexis Carrel, who's a renowned biomedical researcher, and Charles Lindbergh. Yes, Charles Lindbergh of transatlantic flight fame. This book was published in 1938. So it was published almost 75 years ago. And in there they talk about a more biologic approach to the whole area of repair of tissues and organs and replacement. The first meeting that was actually called Tissue Engineering was held at Lake Tahoe, California in 1988. And of course, since then, there have been many, many meetings in the field of tissue engineering. Now, as I think about how industry developed, I'll use some terms that actually, at least in part, should be credited to Michael Lysott, who passed away a few years ago. But the 1990s were the go-go years. Major scientific advances. An industry began to develop. Stem cell technology began to emerge. Everything appeared to be possible. But there was also a lot of hype. The possibilities led to hype. Here you see the ABC science correspondent. I've thought a lot about this, Charlie, and I believe that when historians look back at the 20th century, the greatest scientific achievement will not be space travel or computers, but will be in the fields of tissue engineering and genetic medicine. Well, I think we all realize, all of us in this room, that we still have a ways to go. Another hype was in Barron's, the business magazine, spare body parts, 100 billion plus industry. Well, we're not there either yet, but things are beginning to happen. Now, if we look at the industry in 1994, there were 40 companies, 1,500 people employed. You know, I think the medical device industry, and some of you in this room would have a better idea, but I think the medical device industry employs about 300,000 people. So 1,500 people is a drop in the bucket. There was $246 million in private sector activity. As I've already indicated, compared to the medical device industry, small but still a start. And much of the product development activity was focused on skin substitutes. Some of these are listed here. Dermagraft has already been noted by, I guess it was it you, Tom, that mentioned Dermagraft? And in fact, it has ultimately turned out to be a successful product. Here you see Dermagraft being used on a traumatic wound. What you see in the left picture is the face of a German lumberjack after his face met up with a chainsaw. The middle picture is the same day after his face was pieced back together. And this picture is 6 months later, where yes, you can see the hint of a scar, but still I think it shows how a more biologic approach can lead to significantly improved outcomes. Now, as we got into this century, we entered the sobering years. Private sector activity fell from over $600 million to less than $500 million. There were still a large number of startups, but the number of employees fell from over 3,000 to 2,600. And I think significantly enough, the capital value of publicly traded companies plummeted from 2.6 billion to 300 million. Literally plummeted. So what made it sobering? Well, it was the high failure rate of the clinical trials, a threadbare pipeline, sales of approved products being less than anticipated, and the go-go companies of the 1990s were suddenly facing bankruptcy. We might use advanced tissue sciences as a case study. They were certainly in there in the 1990s developing skin substitutes. The first one was Transcyte, approved by FDA as a bioengineered skin substitute in 1997. Then there was Dermagraft, a dermal equivalent. 2001 has already been indicated. It used allogeneic fibroblasts derived from foreskin. And scientifically and clinically, it was a success, but the business still had to develop. So by 2002, the company went into bankruptcy. More than half the company was acquired by Smith & Nephew, a company that's here today with us. But by 2005, Smith & Nephew announced it was getting out of the skin substitute business, although not out of tissue engineering. Following Smith & Nephew, Advanced BioHealing acquired what had been ATS's skin substitute business. And as you see here, and I don't have 2011 numbers, but you can see that the revenues for these skin substitutes for Advanced BioHealing went from just under 9 million in 2007 to roughly 45 million in 2008, 85 million in 2009, 147 million in 2010. And in 2011, Advanced BioHealing was acquired by Shire Medical. So what turned out to be a bankrupt company has now turned into a relatively successful skin substitute product. What were some of the problems encountered by Advanced Tissue Sciences? A misestimation of the size of the market and the patient need. Turned out that every skin situation didn't need a tissue engineered skin substitute. There were issues at the business management strategy, which I probably shouldn't talk about in public, but there were some real issues there. The regulatory issues, the cost of clinical trials, the delay in reimbursement, basically the time from benchtop to product. Now beyond 2005, we're back to the future. Within the last 5, 6, 7 years, the industry has rebounded. The research continues to advance seemingly at an ever-accelerating pace. With cell source remaining a critical issue, stem cells are looked at as a major part of the solution for the future. There is a realization, however, that regenerative medicine needs to be more than simply stem cells. So if we look at that period, 1995 to 2007, you can see the $250 million that I mentioned earlier, all was in the development stage, nothing commercial. By 2000, it was over 600 million, a little in commercial revenues, but mostly in development stage. A little drop off by 2003, but by 2007, and the latest that we have really complete information on, a significant rebound with half of the 2.4 billion being in commercial revenues. Also, if we look at cell source as an important component of regenerative medicine, back in 2007, 63% of the companies were pursuing an allogeneic cell strategy, only 37% an autologous strategy. Now, as we move forward, clearly repair and regeneration has become front and center. And a lot of this is because of the biological complexity of the tissues and organs of interest, which suggests that repair and regeneration may be more advantageous. And this leads us to the fact that we need to think about how the functional characteristics of a cell is regulated, basically orchestrated by what I call a symphony of signals. And therefore, when we think about a strategy of repair and regeneration, we really need to deliver the necessary signals, cues, at the right place at the right time. That might involve cells, and it might not involve cells. If we can put the right signals in there without cells, that would be a successful strategy. Here, I believe this is a Bob Taylor slide, and I see him sitting somewhere in the back of the room. One can think about how we might mobilize cells from the bone marrow to create repair, regeneration in various tissues and organs. Also, in terms of what I call nature's orchestra, again, the functional characteristics of a cell, including its fate, including the fate of stem cells, is orchestrated by a symphony of signals. This is true of all kinds of cells. What are the instruments that make up this orchestra? Soluble molecules, cell-cell contact, substrate, extracellular matrix, and the mechanical environment. Now, in terms of stem cells, I'm not going to go over this in any detail, but I think we all are aware of the fact there's a variety of stem cells, but maybe you didn't catch it on the news this morning, but the iPS cells are the subject of this year's Nobel Prize for Medicine and Physiology. And certainly, even though I put question marks here because I think there's still a lot to be learned about iPS cells, it has been a major advance, and one can consider it really to be a major breakthrough. If we look at stem cell companies, and again I'm going back to 2007 data, and these are worldwide numbers, 61% of the stem cell companies were focused on adult stem cells, 27% on cord blood cells, and only 12% on embryonic stem cells. And I think if we were to look at the US, the percentage in embryonic stem cells would be even less. If we look at the stem cell market, and this is more recent data from Robin Young Consulting, they project it to increase by 2020 to $8 billion. This should be $1 billion. So if we look at where we are now, relatively small, but a significant increase to $8 billion by 2020. If we look at where that $8 billion market is going to be, orthopedic indications seems to be the largest. Bob Goldberg, I just can't figure out why. Maybe it's the easiest thing to do.
You finally learned.
Still don't believe it, but certainly nerve repair, cardiovascular, diabetes therapy, anti-inflammatory approaches, and although it says dental 0%, obviously there's a small sliver based on Robin Young's analysis in terms of the dental area. A very critical thing, something that we're interested in here, and Todd McDevitt is really providing the leadership in this, and he'll be talking to you later, I think today. But that is, in terms of stem cells, how do we move from benchtop stem cell biology to what's needed for cellular therapies in other applications based on stem cells? Clearly, to get regulatory approval, number one, we're going to have enough cells. Number two, a quantitative systematic approach. Number three, quality control. And we actually have a training grant from NSF to train students in this area. As we think about that process and the design and optimization of that process, we need to really more carefully assess the relevant process variables, how to control these key variables, robust strategies for the interrogation and evaluation of the variables affecting the process, and procedures to test the many different variables that impact the process. Now, we think about translation into the clinic. Obviously, in the US, many states, and worldwide, many countries are focused on the translation of regenerative medicine from the benchtop into therapies for patients. But as I always like to say, the operable word is not simply patients, but having patience. For the timeline from the benchtop to the clinic can be quite long. Bringing a disruptive technology to market takes a while and requires patience. If we look at what's out there, with the exception of the skin substitutes, the other things that have made it to market are largely acellular. This includes Medtronic's infused bone graft approach, also SIS, small intestine submucosa, which is simply a matrix. These are acellular products, and those have been easier to get to the market than cell-based products. But things seem to be going in the right direction. Here you see genetic and engineering and biotechnology news, tissue engineering revenues on the rise. Here we see sort of a pipeline picture that was put together a couple of years ago by one of my friends who's a business analyst in Boston. And although I'm not going to go over every point here, what you're seeing is here the pipeline going from R&D to preclinical studies to phase 1, phase 2, phase 3, ultimately into the market. And here you see the different applications: orthopedic, skin, vascular, neuro, other. And a variety of different approaches: cellular, scaffolds, gene therapy, and so forth. The important thing is that there's a lot of things in play in industry. Now again, this data is almost 2 years old. Some of these things have moved through the pipeline another step or two, but there's a lot going on. But the things that are in the market basically are in the orthopedic or skin area with a few other examples. But there's a lot that's coming along, and that bodes well for the future. There's also been a hope for the use of embryonic stem cells. Jaron actually had a trial for spinal cord repair. The first patient was enrolled here at the Shepherd Center, but in fact, they shut down that trial. That was a business decision. The initial clinical studies actually seemed to be promising. They were phase 1 safety studies, but still promising. But there is an ongoing trial by ACT, Advanced Cell Technology, using cells derived from human embryonic stem cells for degenerative eye disorders. So we're beginning to see some movement in terms of the use of human embryonic stem cells. Obviously, regenerative medicine still has a lot of challenges: cell source, differentiation. Do you use undifferentiated stem cells? Do you use progenitor cells? Do you use fully differentiated cells, scaffolds, extracellular matrix? Depending on what you're doing, you may need innervation, vascularization, the whole issue of immune acceptance. Preclinical animal models, one might suggest that we really don't know yet what the right animal models are for what we're trying to do in terms of cellular therapies. Certainly clinical endpoints for safety and efficacy need to be better defined, regulatory reimbursement, and educated physicians and payers. Now, I put this slide up here because I think it's important. This is a report issued roughly 2 years ago now by the National Academies, where it talks about the essence of the new biology is an integration of the many subdisciplines of biology, and then the integration into biology of physicists, chemists, computer scientists, engineers, mathematicians. This to create a research community that can tackle the broad range of scientific social problems. So here you see the scientific integration leading to a deeper understanding, leading to the ability to address society problems: health, environment, energy, food. Now, I thought I would close with a few slides from a recent study. NIH, NSF, and NIST funded a study to do a global assessment of stem cell engineering. By that, the interface of the engineering world with the world of stem cells. I was asked to chair that committee and selected the panel to work with me, with one of them being my colleague here at Georgia Tech, Todd McDevitt. The idea was to compare U.S. R&D and status with activities globally, identify the gaps, the barriers, identify the major innovations emerging, this to guide research investments, and to identify opportunities for cooperation and collaboration. Again, what is meant by stem cell engineering: the interface of engineering with the world of stem cells, everything from basic research to models, tools, enabling and scalable technologies to stem cell biomanufacturing and the development of stem cell-based products and applications. Schedule of activities: back June 2011, we had our kickoff meeting We spent a week in Asia, basically in Japan and China. There was a workshop in Korea in January this year, trip to Europe at the end of February, where we visited 7 different countries, a final workshop at NSF in May, and the draft report. Basically, we have a draft of the final report, although we're still fine-tuning it. But in that report, we certainly identify some future opportunities for engineers in this world of stem cells. We believe that engineering with its quantitative systems-based approach can contribute much more to basic stem cell research than it has to date. This includes the analysis identifying the components of highly complex cellular systems and providing an understanding of how these work together. The development of improved screening techniques leading to a better understanding of basic stem cell biology and improvements in culture and differentiation systems. In vitro systems benefiting from being engineered to be more physiologic, and this includes organ-on-a-chip models, and both DARPA and NIH are putting a lot of money into organ-on-a-chip models. Engineered in vitro tumor models leading to a better understanding of cancer. That's an area that hasn't gotten a lot of attention, but there's some things now beginning to be done. Computational models, increasingly important to achieve a better understanding of the biology and to integrate into the design of the bioprocessing systems that incorporate feedback control. For stem cell biomanufacturing, a need for further advances in culture system, technologies for real-time monitoring, and process automation. Basically, technology development to enable cellular therapeutics. So, today, in terms of regenerative medicine, this century really represents a new start. The research continues to advance across the broad field of regenerative medicine. The industry has rebounded from the sobering years and is rapidly developing. The promise of addressing unmet patient needs is on the verge of being realized. Still, we have a ways to go, but I think this promise really is on the verge of being realized. Thank you very much.
Thanks, Bob. We have some time for some questions, so any questions from the audience? Maybe, Bob, I'll start with one. First of all, thank you very much for that great historical perspective. One of the things I think we heard actually a couple of the companies say is some of the companies are cutting back in R&D in general, and then when they're being selective, regenerative medicine is one area that some people are cutting back on. So I just wondered if you could comment on maybe the importance of the academic industry partnership to sort of bridge this gap as we're waiting for these technologies to mature.
Well, particularly in the context of the comment you made with companies cutting back on R&D, in particular long-term R&D, seemingly putting their money into the D part of R&D, wanting things that are going to be out there as a product in the next 3-4 years. I think for regenerative medicine to thrive, there's going to have to be continued strong academic efforts, but I think there really has to be also partnerships with industry. And even though industry may not want to put a lot of money into regenerative medicine at this point, still I think it's an area where some investment by industry into academic partnerships will go a long ways, not only in moving the field forward, but in helping the company ultimately realize what they might do in regenerative medicine.
Can you say a few words maybe about what impact inter-individual variation has on the acceptance of medical device interventions? I mean, what's the role of personalized medicine in the future of regenerative medicine?
Well, clearly personalized medicine seems to be the buzzword. When I think of personalized medicine, I don't think of what we might call truly personalized medicine. I think of it as addressing subsets of patients as opposed to one size fits all. And I think that will also be true in regenerative medicine. And as we learn more about how the genetic makeup of a person may enhance or not enhance a particular approach. I think that will be important in all areas of medicine, but certainly in regenerative medicine as well.
Other questions? I'm going to ask you to make a prediction. I know how you love to predict the future. What do you think is going to be the first blockbuster regenerative medicine product?
I think the first area where regenerative medicine, and I'm using that term in a very broad sense, will have a major impact is actually in this area of organs on a chip, which will lead to pharma being able to do many things in the development process using human cells that are in a three-dimensional configuration such as they are in the body, and to better test the drug, initially the toxicity issue, but ultimately the effectiveness of the drug. And I think this will lead to fewer animal studies being needed. Ultimately, there will also be a need for animal studies. But it's a big leap to go from animal studies to putting something in a human. And I think having these human cell organs on a chip will be a major advance.
Okay, let's thank Bob.